The influenza virus. Influenza is a ssRNA virus with segmented genome. It is transmitted through aerosol or body fluid, highly infectious. This is the concept map for the influenza. We're going to begin with the symptoms. Can begin 1 to 4 days, includes chills, fever, sore throat, muscle aches, severe headaches, coughing, weakness, generalized discomfort, nausea, and vomiting, and may lead to pneumonia. It can also be fatal in children and older adults. The rate of infectivity, 5 to 15%. Its mortality rate is also 0.1%. Most recovery occurs in 1 to 2 weeks. The yearly death rate internationally from seasonal influenza is 250,000 to 500,000. Next, we're going to go over is the life cycle. It is intercellular. We're going to begin with attachment. Attachment to a receptor on a target cell expresses two surface proteins, HA or hemagglutinin, necessary for the entrance into the cell by binding to glucine receptors on the surface of the respiratory tract epithelium. The other is the viral surface neuraminidase, NA, is an enzyme necessary for the release of new virions from infected cells. The term tropism is the range of host cells that a particular virus will infect. The next phase of the life cycle is penetration. It's the entrance into the cell by endocytosis, uncoating release of viral nucleic acid through uncoating. Next is replication. It's a synthesis of mRNA and viral proteins. Next is assembly, formation of new virions, and lastly is the exit from the cell, which will lead to either total lysis or budding, where it's cellular death, which then releases to restart the process, or it buds off the infected cell. Viral ssRNA is transported to the nucleus of a cell where transcription and replication occur using viral RNA. Viral proteins assemble in the cytoplasm to form matrix around viral genome. The virion buds from cellular surface. Next we're going to talk about is the eluding from the immune system. Influenza virus eludes the immune system by changing viral surface antigens, called antigenic variation. Antibodies against HA and NA are responsible for protection against the virus. This occurs through antigenic drift or mutations. There's two primary strands, influenza A and B, which infect humans. We are yearly vaccinated is a trivalent mixture of inactive proteins from two influenzas A subtypes and one influenza B subtype. B mutations are much slower than A mutations. Currently there's H1N1, H1N2, H3N5. These are the primary causes of influenza worldwide. Influenza A can also infect birds and mammals, which leads to new mutations from within influenza, which humans can then later contract as well. Viral pathogens bypass many defense mechanisms by hiding within cells, away from normal inflammatory or immune response. So during the process of attachment and penetration, when they're endocytosed by the cell, this is how they elude the immune system. And that is the influenza virus concept map.